Continue on with the CNM Nutrition Seminar Series. So I'm really happy to introduce Dr. David Huyben today. He has been with our department since June 2020 when he was hired on as an assistant professor in fish nutrition. Fun facts about Dave. So he's Dutch Canadian and lived in Europe for 8 years. His daughter was born in Scotland and his dog is from Sweden. So I think interesting, diverse backgrounds there. So today, Dave is going to introduce us to his research program he's going to talk to us about the fish diet and the microbiome. So Dave, really happy to have you. Thanks for joining into our seminar series, and I'm going to pass the floor to you.
Great. Can you
Yes.
see that? I'll start at the start. Can you see that okay?
Yep, you're good.
Great. Thanks for that introduction, Jen, and thanks for having me for the CNM seminar series. And yeah, we'll talk more about fishy business today, but I'm talking about diets and gut microbiomes, and it really does apply to a lot of humans and production animals as well. So there's a lot that is applicable to other research going on at the university. So just a brief agenda, what I'll talk about today, a little bit about me, and I know quite a few of you online, but there's some that I don't as well. And I'll talk about a background, what the microbiome is, and a couple studies from my postdoc, working with Atlantic salmon, and a couple other studies that I've done in the last 2 years being at the University of Guelph. Some conclusions and also talk about some future plans. So I did my master's at Guelph. That was started in 2010 with Rich Macchia. And that was mostly looking at bacteria filtration systems for land-based aquaculture. And then I did a PhD in Sweden at SLU, looking at using yeast as a protein source, and that's where I learned kind of more of a training on gut microbiome research. I did a 3-year postdoc at the University of Stirling in Scotland, looking at omega-3 fatty acids and hypoxic stress on Atlantic salmon. And then, like Jen said, I started a couple of years ago, and I really do focus on the gut microbiome, but I also do a lot of fish nutrition as well. So I wanted to start with a couple of terms, just to make things a little bit more clear. You probably heard these two terms being used a lot, the microbiota and the microbiome. They do have different definitions. The microbiota being more the community of microbes that are there, if it's bacteria, fungi, viruses, or a whole mix of them. And the microbiome mainly relates to the genome, more looking at the function of those bacteria or other microbes that are there. And there are several different types of microbiomes, not just in the gut, but there's also a genital microbiome, oral microbiome, and even skin microbiome. There's basically just microbes everywhere, and they're not all pathogens. They're actually beneficial to us and have helped us for many, many, many centuries. And I'm not just talking about humans. There's also microbiomes kind of on the surface and in the gut and the mouths of different production animals. I do focus on fish, but I also work with Julang Lee and Elijah Carey on different microbiomes of chickens and pigs as well at the department. So I have kind of stretched from just the fish microbiome research. But why is it important and why now? It all kind of is based on the Human Genome Project that got started in the 90s, and it took over 13 years to sequence the genome. It's 3.3 billion base pairs. It involved 20 different universities, and it led to a lot of sequencing technologies and advancements. And so now we can actually sequence a genome within a few days rather than it taking 13 years. And so it's really led to an increase in affordability. So it used to cost around $2 per base pair, and now it's way down less than a penny for base pair. And so it's made the kind of average Joe or Jane researcher be able to afford sequencing the different microbes in the gut or skin of different animals and humans. And secondly, there used to be an older technique that was based on culturing bacteria and then taking a colony and doing Sanger sequencing, which is quite slow. And this is based on using a nutrient media, and a lot of microbes don't like a really rich media full of nutrients, and they also can't grow in aerobic environments. So this really selected for less than 10% of microbes that are actually there. They also had to be living, and so this is actually quite generous. A lot of media can only do maybe 1%. So if you're only looking at 1% of what's there, it's really hard to say what the effect of diet or their environment is on that microbiome. It's also low throughput, so you have to make the media, plate the sample, incubate it for a day or two, even more, and then count all these colonies. And then if there's too many colonies, then you have to do a dilution series. So it's really labor intensive. It's not really cost effective, and you don't get a good picture of what's there. Next generation sequencing, also called high throughput sequencing, you can identify up to 99% of microbes. Basically, 1% maybe they're not in the database yet, but that's increasing more and more every day. It's high throughput, and I'll talk about what that means in a second, but instead of just looking at a few dozen bacteria, you can look at millions of them. It's also very automated. There was a problem about 5 or 10 years ago with big data and having maybe too much data to deal with. But now there are automated bioinformatic pipelines that can look at the quality of data, trim it, and make sure that it's right for your data set. And that's becoming more automated every day using Mother or Time2 or many other pipelines. And so this is just looking at the two different methods again. So we could take a sample of fish or any other animal and then plate it up and then sequence it. And you might get 1,000 or a few thousand sequences, and that might be for one or maybe a dozen microbes. On the next generation sequencing pathway, you can actually pool hundreds of samples into one tube and put that tube on a sequencer. And you do this by adding an adapter and index, basically barcoding each sample. And so when it's sequenced, it actually sequences the barcode as well. So you can relay what that bacteria is to which sample. So instead of looking at one or maybe a dozen microbes, you can have 10 million, and this is actually getting up to the billion level, depending what sequencer you use, and you can identify tens of thousands of microbes. So now we're getting a really good picture of what's there, and we can look at the community as well as very specific targeted bacteria as well. Now, for the human gut microbiome, you probably heard in the media a lot that the gut microbiome has been associated with memory or immune function or vitamins, amino acid production, all these different things. Even resistance to pathogens, therapeutics, storage of fat. However, this has mostly been investigated on kind of the human or at least the mouse side, and there hasn't been a lot of research for this on the animal side, especially for fish. Just in starting kind of in 2016, 17, I was part of quite a few studies that map the different microbiomes, mainly the gut of rainbow trout, Arctic char, and Atlantic salmon. And they do have a lot of kind of overlapping phyla. So one of the largest, highest level of bacteria with a lot of firmicutes and then proteobacteria. And there's a lot of other phyla as well, such as tenoricutes. And so similar to humans and other animals, I mostly look at the gut, rather what's in the intestinal wall, so what's inhabiting or setting up there, and then what's in the content as well, so what's going through the animal in the feces. But there's still a lot of work to be done on the gill and the skin microbiome as well. This is just a very complicated figure to show you that the microbiome is very complicated, and there's a lot of different impacts that can happen. So on the host side, different stress level, immune status, different genetics can impact the different bacteria and fungi and other microbes in the gut. For the environment, pollution, water quality, even where they are, can impact them. I mostly focus on the diet, so looking at the composition of the diet, what dietary sources are used, if there's any sort of probiotics involved as well. But there are many different effects. I kind of boiled it down to 4 in particular. So we have the more environmental and dietary impacts, and the host-associated impacts, such as genetics and life stage. And so you can actually modify these groups to change what's in the gut microbiome, the nutrients they produce. And you can improve the production of salmon or other salmonids, such as rainbow trout, Arctic char, lake whitefish. And it can improve the stocking, so both kind of on the fishery side, the stocking from hatchery fish, and also improve farm growth, so increasing the aquaculture industry as well. So we really need to use the gut microbiome and utilize it as that kind of extra organ, as it were, and take advantage of this amazing system. And so there's also pathogen antagonization from a few different mechanisms. Most studies look at probiotics, but it also impacts the normal microbiome. So probiotics can inhibit certain substances, such as mycotoxins, that might impair the epithelium or the intestinal wall. They might interact directly with the host, or change it indirectly by modifying the microbiota, which in turn affects the host and its defense. So different immune cells, such as interleukins, that target different pathogens. So and also probiotics can directly exclude pathogens as well. So there's 4 different mechanisms where the gut microbiome and probiotics can have a beneficial effect on the health of animals. And I like to dabble in many different feed ingredients, so not just fish meal and fish byproducts, but yeast, different bacteria meals, and insects as well. Recently, I looked at brewery waste as a protein source and also a lipid source, and I haven't yet, but I'm looking at microalgae in terms of high EPA and DHA, so those really good omega-3s. Kind of compared to, if you just look at a nutrition, because this is a nutrition seminar, fish meal is kind of the gold standard for high quality protein in the fish industry, and probably a lot of the animal production industries, where soybean meal is kind of that really lower quality protein. And kind of yeast and algae and fungi, such as yeast, are very similar to that soybean meal. So not really high on the potential for protein replacement. However, there's some bacteria meal and also other aquatic byproducts, such as blue mussel. And there's also crickets that are getting up here as well. So this is black soldier fly. It's around 55% crude protein, but there's also crickets that are a little higher. And so that's a huge potential as well. But we're not just looking at a protein alternative. We're also looking at functional feeds. So a lot of these cells can improve the immune response as well as insects and different ingredients. For example, the yeast cell wall. So yeast can be autolyzed to have an extract, which is rich in protein, amino acids, vitamins, a lot of the B vitamins and different enzymes. But the cell wall is really rich in beta glucans and mannan and oligosaccharides, and these act as prebiotics to feed those probiotic bacteria, and they can also have an effect on the immune response of animals, especially there's been a lot of research on this in fish in the last few years. And for insects, there's quite a few advantages. Fish naturally eat insects in the water or even in the air, as you can see by this jumping fish. And we don't use the adult fly, black soldier fly, we use the larvae. And they only take a couple weeks to grow from an egg up to a larvae stage, and they're dried and ground and used in different feeds. So they're very efficient at converting organic waste into high quality feed ingredients. They have a very low environmental footprint. They can be fairly high in protein. There's a couple amino acids that could be better, but such as methionine, but these can be supplemented. It's really rich in saturated fatty acids, such as lauric acid, and that's known to be an antimicrobial. So we're looking more into this. And the exoskeleton of the larvae has a lot of chitin in it, and that can be used as an immunostimulant. Some disadvantage is the cost. It is very expensive right now, but it seems to be coming cheaper and cheaper as the industry increases in scale. And there's also an efficiency of converting that waste into feed that can be improved. But there's lots of research going on that side as well. So the first study I want to talk about is feeding yeast, BW, that's brewery waste, and insects to Atlantic salmon. So this is from my postdoc work. It was in a flow-through marine-based system with 300-gram fish, and we did a 30% exchange with distillery dried grains yeast, brewery waste, and black soldier fly larvae. It was just a 3-week experiment, so 7-day changeover, and we stripped the feces as well as looked at the contents. And this was at the Mackerhanish Environment Research Laboratory at the University of Stirling. And this work was, it's not been published yet, but it will be submitted soon. And what we saw was, in terms of diversity, and what I mean by diversity is having many, many players on the field that do different roles. So if 1 or 2 go down, there'll be others that can take its place. So it's really beneficial to have a very diverse gut microbiome. And when we fed the insects, we saw the diversity, both in terms of evenness and richness, go up. And this is a really good thing to find. And so we're looking at this further to see, okay, is it the chitin or is it the lauric acid? What is improving the diversity? And we saw an increase in bacillus and bacilliaceae bacteria that are known to degrade chitin. So is it just that we're supplying more of the substrate to grow that bacteria, or is this having a beneficial effect on the fish? We also found the yeast had a very different composition of bacteria versus the diet and versus the basal control fish. And the brewery waste was fairly similar to the basal control. And this is similar to another study that I did way back in my PhD, where we saw bacilliaceae bacteria increasing when fed different defatted and full fat larvae. So some larvae are defatted to reduce the saturated fatty acids and increase the protein level. And we also saw increases for all 3 diets versus the fish meal control. So this seems very in line with my previous work. Feeding omega-3s and lipids. So my second study from my postdoc, so it was 200-gram Atlantic salmon in a flow-through marine system. we had high and low levels of omega-3 and then high and low levels of lipid. And so the idea was that if you feed more omega-3s, do the bacteria in the gut react to this? Do they produce more or less fatty acids? And also, does the amount of lipid change the gut microbiome, what's there and what they're producing? It was 35 and 116 days, and we looked at the distal content, so the feces in those fish, and it was at the same research lab on the west coast of Scotland. And this was published last year, where we found that lipid really early on in the study. So we looked at day 35 kind of as a short-term effect. Found that lipid really did change the diversity of the gut microbiome. So those HL, the high lipid diets, had a lot more bacteria in them. But at day 16 or 116, this lipid effect went away, and then we had an effect of omega-3, which is kind of interesting. Whereas if they're fed omega-3, so the diets with the plus, these two diets, they actually decreased in the amount of bacteria diversity that was in the gut. So a bit of a head scratcher, and we found a couple of studies before that adding more omega-3 can actually have negative effects on lactic acid bacteria, and that's negative for the diversity and the health of the gut microbiome. So kind of having that balance of maybe not low omega-3s because they need that for health and growth of the fish, but not adding too much omega-3 that it's negatively affecting that gut microbiome and that gut health. You can also see the mycoplasma, that's a very negative bacteria associated with tumors in the gut, and that seems to be really increasing for the high omega-3 diets. And a lot of the beneficial bacteria such as Streptococcus, Wuerthelia, and Lactobacillus, it's all lactic acid producing bacteria, they're all decreasing for the high omega-3 diets. We also did a additional analysis looking at metagenomics, so not just what's there, but what those bacteria are doing. So we use pie crust to look at functional estimates, and we actually found that when they were fed low amounts of omega-3, they had increases in fatty acid biosynthesis. So the bacteria, if those fish weren't being fed high omega-3s, they were able to produce some fatty acids in the gut. So we know they produce certain fatty acids and vitamins, and it's really interesting that they kind of made up for this deficiency in the diet. It's really cool to see. And there's certain bacteria such as Bifidobacterium and Corneobacterium that really increase. So these two bacteria seem to really take over and produce a lot of fatty acids. But this is based on functional estimates, so we need to do more empirical research and look at the transcriptome and what's actually there. But a good first step. And this is very similar to a study of a colleague of mine did in Chile with wild flounder. And they found very similar effects when they had wild flounder that were fed low amounts of omega-3, they had increases in fatty acid biosynthesis pathways as well. It's very interesting. So in conclusion of the first two studies, we found bacillus and the diversity of gut bacteria increased. So this is a very positive effect, but we have to see if this translates into improved growth or any sort of phenomic metric. And also the lipid affected certain bacteria and increased diversity. So what was going on with that kind of short-term and long-term effect? Is that true for other species, and why is that happening? The effect of omega-3 took longer and reduced diversity. So maybe if we have a high omega-3 diet, we could add a probiotic to offset this. And then lastly, the gut bacteria could compensate for the lack of those dietary nutrients, but more studies have to be done. So kind of 4 conclusions, but also 4 questions, or if not more. So that's fairly common. And now I'll just go into another study that I did at the University of Guelph. We looked at something called eubiotics or postbiotics, which is a fancy new term, which includes prebiotics, probiotics, and symbiotics. But the eubiotics also include essential oils. So these aren't like essential fatty acids. They're rather just the extract or the essence of different plant-based oils, from garlic, cinnamon flowers, even cannabis. And this is growing in a lot of research and a lot of kind of focus, both in fish, but in other human and animal trials. And there's also organic acids that have been around for quite a while. And so that's acidic, lactic, citric acids, and it's basically paired with a salt. And this improves kind of gut pH and integrity. So the third study was feeding rainbow trout around 122 grams in a flow-through freshwater system. And this was done at the Ontario Aquaculture Research Centre. And so we just had a control and then another treatment that was composed of organic acids, so a blend of these 4 different acids, and essential oils, so thymol, valine, and eugenol. And it was a fairly short study looking at histology and the distal content, looking at the microbiome using MiSeq Illumina sequencing. And we found the villi length increased in the proximal intestine. So we see kind of a direct effect on the gut health. We didn't see an inflammation, any change, but we also didn't challenge them with the soybean meal diet. And we're going to do that later on. And we also found that there's some bacteria such as Aeromonas that decreased, and this is a huge pathogen in fish, especially Aeromonas seminicida and Hydrophila, causes furunculosis and some other diseases. And so it seems like it really didn't change the diversity of the bacteria, but it actually targeted specific pathogens, which is pretty neat to see. A 4th study, so this is the final study I'll talk about, is the lake whitefish fed lipids and also insect protein. And this is research that was presented at the Aquaculture Association of Canada conference in St. John's a couple weeks ago. And here's my master's student presenting. So quite proud of her, and really great to go to a conference with her, and I haven't been for 3 years, so that was really great to attend. So why lake whitefish? And what is a lake whitefish? They are a salmonid. They're more of a deep water fish, and they are native to the Great Lakes, so that's really great. There's been kind of not a lot of buy-in for producing land-based shrimp or tilapia. Even rainbow trout is a naturalized species, so they're not exactly native to the Great Lakes. But the lake whitefish are. There's one commercial fish farm on Manitoulin Island out of 11. And there's a lot of advantages to them. They do survive really well in deeper waters, and they really work well in that kind of big net pen environment in the Great Lakes. And there's been a recent collapse of the wild fishery. So around Manitoulin and Sudbury, Northern Ontario, they eat a lot of lake whitefish in their fish and chips, and just generally. And so they really rely on them. And so the wild population has collapsed. And so one solution is to farm them, which is where a lot of the farming around the world comes from, is to relieve some of that pressure on those species and make sure they recover, and while we eat them from the farming system. It's also very important for the First Nations that have been relying on the fisheries for centuries. And it's also very high in omega-3 fats, even higher than rainbow trout and Atlantic salmon. So humans who eat them can get those beneficial effects for really good heart health and brain development from eating high amounts of omega-3s from lake whitefish. They are kind of a, as you could think, kind of a white silvery look, and they actually look blue underwater. There's a group, we took the departmental staff to the research station in Alma last week, and if anyone saw them at the last kind of tanks, they look very blue underwater, but they are very white silvery look to them, very long. So we did an experiment last year in the flow-through system at the Ontario Aquaculture Research Centre in Alma, and we had high and low levels of lipid and insect proteins, so a similar 2x2 factorial design, plus a commercial control. They actually have been feeding them a rainbow trout diet because there has been no commercial diet developed for the lake whitefish, and this might be leading to reduced growth and poor feed conversion ratios. It's a 116-day study, and we looked at their weights every month, and we sampled their liver and did RNA extraction and qPCR, looking at the differential gene expression. So we had quite a fun time making feed. My master's student Rebecca Lawson was involved as well in this. I'll show you just the qPCR results, and we're just waiting to get the microbiome results as well from that. Had a URA student, Liz, and other master's student, Carmi and Huyben helping on this project. So it was really fun. Quite a few challenges to do an experiment during COVID, but we got it done, and I think it went really well. And for weight gain, we found the blue water commercial control diet was the highest in terms of growth, followed by the high protein and high lipid diet. However, we found the VSI was also the highest for the commercial control, where it was lower for the HPHL diet. So this meant that it would have less kind of fat in the gut, and the high protein, high lipid diet would be better for both the farmer and the consumer, because they're putting more of that weight on the flesh rather than on the internal organs that are eventually degutted. Looking at the gene expression, we did find lower levels of heat shock protein 70, and this is involved in cellular repair. So we found that both the control diet, the blue water commercial control, and the high protein, high lipid diet had lower levels, and this could relate to lower stress level and improved efficiencies. And in terms of growth, I just kind of mentioned that earlier. It was more optimal to have that high protein, high lipid diet. And the high lipid diet had about 18% crude lipid in it versus 12. And there wasn't any effect on the innate immune genes. So we looked at interleukin 1, 6, and 10, and a few others. And we didn't see any significant effects, but we only had 5% inclusion of the insect meal. So whereas a lot of the other studies that have found immune effects have 15 or 20% inclusion. So more work to be done on that side. So kind of the conclusions from study 3 and study 4, organic acids and essential oils seem to target specific bacteria, and this is kind of opens up potential to use for an alternative against antibiotics. So similar to what Elijah mentioned in the last CNM seminar, was trying to get away from those antibiotics and using different functional ingredients to improve the gut health and the gut microbiome and reduce disease. However, this study that we did used a blend of both of them. So there's a lot more research to be done looking at individual organic acids and essential oils and see if there's a synergy between the two when they're used in combination. For the FORCE study, that high protein, high lipid diet was optimal, but there's a lot of research that has to be done. What are the limiting amino acids? What is the level of lysine, methionine that should be in the diet for this species? No one's ever looked at that. Even fatty acids, not just EP and DHA, but other fatty acids that are essential, and minerals and vitamins, the whole gambit. Also, what is the impact of genetic selection? So the lake whitefish we used were recently removed from the wild, so they were maybe bred once in captivity, whereas the rainbow trout and the Arctic char have been bred for 30 or 40 years in captivity and selected for different traits such as growth and really efficient feed conversion. So we'll see kind of as the ALMA, and Marcia is involved in this, selects for improved growth performance of the lake whitefish. How does that respond in terms of their diet, and how can we optimize both their genetics and their nutrition at the same time? So future plans, there's a study ongoing right now at ALMA that a lot of us looked at last week, looking at rainbow trout fed different levels of black soldier fly at 5 and 10%, as well as looking at just their insect fat and just the insect chitin. There will be another study in the late fall looking at zebrafish, and this will be done at the Hagen Aqua Lab on campus in the integrated biology department, where they have a broodstock or a whole breeding population of zebrafish. And they're really cool model species because their whole genome is sequenced, and there's been a lot of research over the last 10, 15 years, kind of from human perspective or even biomedical perspective. And so they're really good to relate to farm species as well. And we'll feed them different insects, probiotics, prebiotics, and even organic acids. And this involves 4 HQP grad students, and this is with Neil Caro and Dom Bureau, even other universities such as Laval, and a couple industry companies, Intera that supplies insects, and Alltech that supply the pre and probiotics. There's another study that's ongoing, and we'll look at the antibiotic resistance in Ontario fish farms, and as well as fish farms on the east and west coast. So this is a nationwide project ran by the Fisheries and Oceans Canada, so the DFO, and the Environment and Climate Change Canada, the ECCC, as well as other collaborations with McGill and industry partners such as Cole Munro that own the fish farms, most of them in Ontario. A couple of students involved in that, and we also have an ongoing project with some students with Emma Allen Vercou at the Molecular and Cellular Biology Department across the street, and also hosting a visiting PhD student from my old university, SLU, and looking at the artificial fish gut. So rather than doing a 116-day experiment, can we replicate this within a few weeks in a bioreactor that has kind of constant pH and aeration and inputs of feed, and as well as bacteria, and see how that develops and how that changes based on different ingredients you put in those bioreactors. So that's really cool experiment we have going on. And Emma has developed this for the human microbiome, and we're using it to develop a fish gut model. So that's really cool to see. So instead of just looking at diet, a lot of these future projects looks at location, genetics, even life stage. So looking at not just adult rainbow trout or salmon, but from fry, juvenile, adult, the whole from hatchery to production, and looking at how can we improve the nutrients that they produce and their interaction with these animals and improving both the stocking of wild fish as well as stocking of farm fish in Ontario, Canada, and globally. I just wanted to thank all the all my collaborators, Rich, Neil, Dom, Jen, Emma, Marcia, it's here. Also Graham, Wes, Ann, and Mike at staff at the research station, all my students, Huyben, Carmi, Liz, Junyu, Cody, Rebecca, and all the industry and funding bodies as well. But that's it for me. Hopefully I was okay. 4 minutes under, so that's good.
Thanks so much. Thanks so much, Dave. That was really great, and I love the ugly Christmas sweater.
Yeah, you gotta have some fishy fun sometimes, right? So yeah, we have lots of not a lot, but we try to get together once a semester and have a themed lunch, and so I didn't show a picture, but we had a summer Hawaiian themed lunch with lots of ugly Hawaiian shirts. So that'll make the next presentation, I'm sure.
Nice. Thanks so much, Dave. So that's wonderful. So we have time for questions. So don't be shy, guys. You can put up your virtual hand, or you can just unmute yourself and ask a question. But questions, what do you guys have? Don't be shy. I see a hand. Let me see who that is. Chris, yeah, Powell, go ahead.
Hi, Dave. Great presentation. It's nice to see what you've been up to for the last couple of years. So it seems that a lot of your trials revolve around the use of insect meal. I haven't been in the fish game for a couple of years now, but does the CFIA, has that approved the use of fish meal for, not fish meal, sorry, insect meal for the use of, aquaculture and the fish that we eat?
Yes. Hi, Chris. Nice to see you. Yes, so the Antera is the biggest producer of black soldier fly in Canada. They're based out of BC, but they have offices across the country. And they've been approved at least for 3 of their products, I think. The black soldier fly, defatted black soldier fly protein, the grubs. So they actually sell that to pet stores for lizards and things like that, and also their oil, and they've been using it in human ingredients as well. So it's been approved for animal production, including fish, and they also have some approval for used in human, like protein bars and things like that. Black soldier fly flour, and there's been a few other companies such as Aspire that are doing like a cricket meal, even like cricket chips and puffs that are cheese flavored and all kinds of things. So yeah, I think it was, I can't remember exactly what year. I think it was maybe 3 or 4 years ago. It was approved for sale to the human food chain rather than just the animal food chain.
Okay, great, thanks.
Okay, other questions? Be shy.
Maybe if we have this in person, eventually I'll get some cricket chips and things that we can snack on.
You know, the first CNM seminar that we did, it was Mike Steele's lab. I don't know if he's still online, but they made pancakes live, and with oh, I'm forgetting the word, but anyway, but they made pancakes live in 141, which was our original plan to bring snacks. But
Nice.
then COVID hit almost immediately after. Oh, there's Mike. Yeah.
Those were colostral pancakes.
Yeah, that's right. The word was missing in my on my mouth, but yeah, it set a really high bar, and then we all got shut down by COVID. So,
Yeah.
So I expect cricket chips at our next in person one day.
Yeah, sounds good. We're trying to organize. There's another company we collaborate called Entemo, and they're out of Peterborough, and we're trying to get kind of class trip to their farm. They converted 3 old chicken barns to cricket farms. They also do super worms, and yeah, pretty cool to see. And we haven't been there yet, but I'm excited to go and try some of their products as well.
Nice. Yeah, there's another hand up. Yeah, you can go ahead.
Hi.
Hey, Basim.
Yeah, hi. It was really interesting, and it remembered me my first year of working on bioinformatics, because I started with metagenomics and with Bacillus species. So
Oh, cool.
it remembered me 10 years ago. Yeah, when I started. So I have a question. You know, I'm from Tunisia, so we have the Mediterranean Sea, and so. Did you do any comparison with other places around the world? Is it, for example, here I know that there is winter, so the temperature on the water is cold. I mean, colder than in other places around the world. So, do you think that the metagenomics there or the microbiome will be different depending on the place where the fish are? Or it's maybe related to some other things rather than the environment where they are?
Yeah, very good question. There's been a couple studies on that, specifically more looking at salmon in Canada versus Ireland versus Scotland versus Norway, and it's a study by Llewellyn in 2014. I referenced it a couple times, and they found that even if it was a different location, they had almost the same microbiome, but when it came to different ages, then that seemed to really change what they found. So there seems to be some sort of hierarchy that if it's their genetics or life stage at that time really puts pressure of what bacteria there are at that time and what diversity. So usually fish are kind of opposite to humans. As we get older, our gut microbiome gets more diverse, more rich, but fish, it gets reduced over time. And so that seems to, something happens and they can't filter as much bacteria, or they just kind of let it go. And so there seems to be an age effect. And if it was a different freshwater versus marine, seems to be saltwater versus fresh, that kind of really has a huge effect on the microbiome. And even different species, like it's fairly similar, like I was showing with salmonids, but if it was like a sea bass versus Atlantic salmon, they would be quite different. But they're very different temperatures, so that makes sense. Yeah,
Okay,
good question.
thank you,
No
Dave.
problem.
Okay, we have time for one last question, if there is one. This is your chance. If not, I'd like to thank Dave very much for giving the talk today, and I'd like to thank all of you for coming. We will continue the CNM seminar series in September. I believe it is John Kant's lab's turn in September. And I think the plan will be that we try to offer this in a hybrid format going forward. So we'll move into 141, but we'll still offer the online option because I think we've gotten a really nice diverse group of attendees from outside the university as well. So I think it's nice to keep it open that way. But just stay tuned for the next invite. And if you'd like to join us in 141, we'll be in there in person, and otherwise we'll see you online. Thanks again, Dave, and thank you, everybody, and have a good last few days before the end of the semester and the big start to the fall semester. Thanks, guys. Have a good one.
Thanks, everyone. Have a good day.